Hi, my name is Carrie Wong, and I'm a Senior Study Director in Discovery and Translational Sciences. I would like to share with you how global metabolomics can be useful in identifying biomarkers in clinical trials. Today's case study will be in the area of NASH R&D. A challenge of studying NASH is the lack of robust blood biomarkers. Current strategies to assess this disease depend on imaging and biopsies, which can be expensive and impractical. In the study I am talking about today, scientists sought to identify plasma biomarkers in a phase 2 trial testing an ACC inhibitor in NASH patients. ACC is a rate-limiting enzyme in lipogenesis. Global metabolomics was performed on plasma from these patients. All of them received the novel molecule, and response was assessed by MRI. A subset of metabolites, known as acylcarnitines, were found to be decreased in the responder group. Acylcarnitines are useful surrogates in assessing fat oxidation. Relative changes from baseline in ACC levels between responders and non-responders were significantly different at week 12 in the dosed group. A targeted panel of 22 acylcarnitines was then developed. Further analysis demonstrated that 4 acylcarnitines had good area under the receiver operating curve for identifying MRI responders at week 12. These data provide an example of how global metabolomics was useful in developing a panel of novel biomarkers of response that could be mechanistically linked to ACC inhibition and liver fat content.